Alright guys, let's continue our discussion on testicular cancer by talking about our germ cell tumors. I'm going to put this little purple squiggly next to our germ cell tumors to remind you guys that 95% of all of our testicular cancers are going to arise from our germ cell tumors. You'll remember in our video on testicular cancer, we talked about our 95% arising from our sperm cell progenitors, which was this purple population of cells that we talked about that are different from the stromal cells, which were our Sertoli and our Leydig cells, which we denoted in orange over here. So now that we're kind of oriented to the cell type that we're talking about, let's get into this table where we're going to talk about the specific things you need to know about each of our type of germ cell tumors. We're going to start out by talking about the epidemiology and presentation of each tumor type, and then we're going to move on to talk about some of the lab values that you might expect and some of the histological findings of each of these tumor types. So let's start off by talking about our seminomas. And seminomas are going to be the most common type of testicular tumor. They present as a homogeneous testicular enlargement, and this is going to be a painless tumor type. So in a vignette, this would be a patient that notices a painless lump on their testicle. This should not be painful. And then in terms of the prognosis here, these are going to be malignant tumors. However, they take a while to metastasize. So there is some time that you have to diagnose this before it goes on to full metastasis, which means that they have a really good prognosis. Like I said, this is going to be the most common testicular tumor, and then important here is that this type of tumor does not affect infants. So you will only expect to see this tumor type in aged toddlers and above. No babies here. So talking about the tumor characteristics, and it's important to know that this is the male analog of ovarian dysgerminoma. And remember when we talked about ovarian dysgerminoma, we talked about the classic fried egg cells, and these are described as a vacuated watery cytoplasm with a central nucleus. And so this is going to be a large cell in a lobule of watery cytoplasm, classically described as fried egg cell. So let's take a closer look at this fried egg cell so we can get a better idea for test day. I'm going to outline a classic one here so you can notice what I'm outlining is a cell that has this white watery cytoplasm. This is like the egg white, and then that purple nucleus in the center is like the yolk, and this looks to me like a fried egg in a pan, and that's a great way to recognize this on test day. In addition to the fried egg cells, we can expect to see increased placental ALP, and it's notable that this is a radiosensitive tumor. So in addition to radical orchiectomy, if this tumor had metastasized at all, the metastasized tumor is going to respond really well to radiation. So that's really great, especially because this is a tumor with a really good prognosis. Now, let's move on to our next tumor, which doesn't have such a great prognosis, and this is like what I think of as the meanest testicular tumor. This is going to be our embryonal carcinoma. And the reason why I think of this as a mean tumor is because it is painful, it is hemorrhagic, and it's associated with necrosis and a poor prognosis. So painful mass, hemorrhage, necrosis, and then these are malignant, and as opposed to seminoma, they're going to metastasize very early. So they have a poorer prognosis compared to our seminomas. Additionally, these can be mixed with other tumor types. So that's an additional aspect of this tumor that's important to know. And you can differentiate a pure versus a mixed tumor based on other lab values, which we'll talk about in a second. So when we talk about the tumor characteristics, it's going to have glandular and papillary characteristics on histology. So let's take a closer look at this histology and what we're looking for. And I want to actually compare this to seminoma because they are so different. You'll remember in seminoma, the tissue was very uniform. It was all these fried egg cells just kind of lined up nicely. Whereas you'll notice that in our embryonal carcinoma, we have this like hemorrhagic necrosis occurring here, and this is associated with nucleus that are overlapping. So you can appreciate that these nuclei are all overlapping on top of each other. We have these associated red blood cells which represent the hemorrhagic necrosis, and then another finding you're going to see here is clusters of cells surrounded by white space on three sides, like this example here. And it's important to differentiate this from seminoma, which also has a lot of white cytoplasm around it, but you'll note that in seminomas we only have one nucleus surrounded by white cytoplasm, whereas in the embryonal carcinoma here, we have like 3, 4, or 5 nucleoli all within this white cytoplasm, and you're going to have all this disorganized hemorrhagic necrotic tissue, and that's how you're going to differentiate this really aggressive, mean, malignant, bloody, painful tumor from the more benign, non-painful seminoma that has a good prognosis. So let's move on now to talk about our teratomas. These are also an analog that we talked about about in the female pathology unit. So let's go ahead and talk about it in the male. So in our female patients, these were always benign tumors, whereas in the male population, these can actually be malignant in our adult male patients. So differential presentation with age and sex. In our adult males, these can be malignant or benign. So I'm going to note that they're malignant here in the adult male as a possibility, whereas in our pediatric males, these are always going to be benign, and in all of our females, these are going to be benign. So basically for teratomas, if you have an adult male, you want to have a higher suspicion that this could be malignant. Now, in terms of the description of the tumor itself, this is going to be very similar to the female analog. It's going to be a cystic mass, and important here is to note that there's all 3 germ layers. So I'm going to highlight the 3 in red because that's really important. We're expecting a heterogeneous mass composed of ectoderm, mesoderm, and endoderm. So all 3 embryological germ layers. And another way that they may hint at this on test day is by describing a lot of different tissue types. So they might say that it's composed of hair, teeth, and sebaceous tissue, a mixture of different tissue types. This is key to the teratoma. And you can also appreciate this on gross exam. If we zoom in a little closer here, you'll notice that this teratoma has distinct regions of tissue. So here in the periphery, we kind of have this darker tissue type, and then we have this really nodular tissue type up here, and then we have a more uniform tissue type down here towards the base. This is representing all the tissue layers in harmony within this teratoma. Let's move on now to talk about our yolk sac tumors, which are also known as an endodermal sinus tumor, and this is going to be the most common tumor in our children below 3 years of age. So as opposed to our seminomas, which spared our infantile population, our yolk sac tumors can actually affect the much younger kids, and this is going to be a malignant and aggressive tumor, and it's the male analog of the yolk sac tumor that we saw in our females. On gross exam, this will be described as a yellow and mucinous tumor after radical orchiectomy, and then on histology, we expect to see what are called primitive glomeruli, also known as Schiller-Duval bodies. So let's take a closer look at what a Schiller-Duval body is, because I always had trouble recognizing this as a medical student, and it's important to note that you're going to look for like this central shard-like eosinophilic inclusion surrounded by cells with a lot of purple nuclei. So you can appreciate a great example here. Here's this like really discrete eosinophilic shard. This represents the blood flow of the primitive glomeruli, and then you have these purple cells all surrounding it kind of in a circular pattern. That's how I recognize this one on test day. And then additionally, in terms of lab values, you can expect to see increased AFP on this tumor type. Finally, let's wrap up by talking about choriocarcinoma, which is going to be a malignant tumor with hematogenous spread, which means that it can easily spread through the bloodstream. And we talked about this a little bit in our talk on molar pregnancies, because this is a complication of molar pregnancies. However, this can also arise as a testicular tumor from the germ cells of the sperm. And so what happens here is we get metastasis to the lungs and the brain, and these are classically described as cannonball metastases. So there's these big ball-like metastases that you can see on X-ray in the lung or on head CT in the brain. And then these can be associated with gynecomastia and hyperthyroidism, and this is due to the fact that these tumors just pump out a ton of HCG, which causes these hyperthyroid type effects. And if we were to look at the cellular histology here, we would expect to see disordered trophoblastic tissue, and we know that trophoblast is made up of both syncytia trophoblasts and cytotrophoblasts. So I'm going to underline the syncytia trophoblastic tissue in purple and then our cytotrophoblastic tissue in orange. And normally during development of the trophoblast, these two tissue populations are distinct, so they're separate layers. However, you will notice that in this tumor type, we have disorganization of these two structures. So in our cytotrophoblast, which is in orange, we have it kind of lacing through this lighter tissue here. It's all the light eosinophilic tissue, whereas our syncytia trophoblast is this darker purple tissue, and you will also note that it kind of laces through disorganized. We literally have cytotrophoblastic tissue inside of syncytia trophoblastic tissue. This is no organization whatsoever. Normally this should be two distinct layers, and this is a great way to denote this type of tumor on histology. So if we were to also look at our lab values here, we would notice that this is an increased HCG lab value. This is going to be severely increased because just like in molar pregnancies, this tissue type is just just pumping HCG, and this is going to be a great way to clinch this type of testicular tumor on exam day. So to review, it's important to know the different types of testicular tumors that arise from germ cells. Remember, these are 95% of all testicular tumors. You need to know the histological findings of each of these as well as the lab values associated with them. We're going to talk about the last 5% of our testicular tumors in the next video. Can't wait to see you guys there. Bye!